Let's begin cycle 3 by characterizing the genome. When I say characterizing, we're going to characterize it using numbers, actual values for these chromosomes. So we'll talk about the ploidy, the number of unique chromosomes, the copies of the genome, the size of the genome, and put actual numbers to various different species. We will use the human as an example because it's easier to learn it with that example, but wherever we get a chance, we will talk about other organisms. So here's a slide showing you 4 different karyotypes of different organisms: the human, the frog, the sunflower, the saffron finch, which is a type of bird. This is a karyotype. A karyotype, if you recall from last lecture, is an image that is taken by scientists. And the important thing to note here is that this image is taken from chromosomes during metaphase. So that's really important to know because you need to understand how the chromosomes are during metaphase, i.e. there are sister chromatids, right? It will help you understand how many copies of the genome are there because it has already gone through S phase. It'll help you understand the ploidy based on centromeres. It'll help you understand what the karyotype is telling you because you know that this picture is taken during metaphase. This is when the chromosomes are condensed and they're just able to isolate it better at this stage. So they isolate it and then they line it up from largest to smallest and they take a picture. So there are karyotypes for almost all organisms in the world. It's published, you can just look it up, and it gives you a lot of information. If you look at the human here, you can see that it has 23 unique chromosomes, and it's a diploid because you can see a homologous pair. Each one of these is a homologous pair. In the case of a sunflower, this is a tetraploid. So most plants, or many plants, are polyploid. So they have polyploidy. And there's an advantage point for this. Plants didn't just, randomly evolve to have many sets of unique chromosomes. It's because it helps them adapt better. It helps them adapt better. Imagine a plant is just still, right? I mean, it really can't have locomotion and move based on its environment. So it needs to just really adapt. And a lot of mutations then can be masked. You can mask the mutations. Even us as diploids, right? With these recessive genes that we have, we can often mask them with the dominant. So imagine with the polyploid, you can do a lot more. You can breed a diploid watermelon with a tetraploid watermelon, come together to make the seedless triploid watermelon. I prefer the triploid version without the seeds. So understand that they're polyploid for a reason. We won't touch on the details, but that's a really generic reason for you to understand why they're polyploid. The frog here is a diploid, only has 12 chromosomes. You can see from this karyotype. And the bird, these are all the chromosomes of the bird, and these last 2 are the sex chromosomes. So understand that when you look at the karyotype, you can really get a general sense of the genome of the organism. So why do we want to understand and learn about the genome? What is the importance of the genome? Why do we care to understand this? I'm putting this as a lecture because this is critical. If you want to understand an organism and how it's behaving and what it's doing and why it's doing it and predict things for the future, predict things for for what could happen with respect to disease or with respect to anything, the information really is in the genome. The genome codes for everything, right? Your DNA codes for everything. So even if it's a non-coding region, meaning it doesn't code for an RNA or a protein, it still has very important functions. Those non-coding regions play a pivotal role, and they help the coding region to actually produce that product, which is the RNA and the protein, which ultimately performs a function in the cell. So if you understand the genome, you can understand the organism. You can understand your ancestry. You can understand if, what the probability is that you'll get a disease. You can understand what mutations you have that already lead to you having the disease. So that can be used for targeted therapy. In other words, let's make the therapy very, very targeted to this individual, right? So the genome has very important information. So if you want to study life sciences and you pick up a species, the first place you have to start really is in the DNA. You can also use this genome quantification to compare different species. Most scientists don't study all species, obviously. They study a group, or they can even just focus on one. But you need to make comparisons to understand, this species can do this. Why is that? Why is it that it cannot do this versus this? And when you make that comparison, you get a better understanding of the species itself, or even evolution. If you look at our DNA today and what it was 100 million years ago or even 50 million years ago, it's very different. So is our genome evolving? Absolutely, it is. It's evolving very slowly, though. So you're not going to see drastic changes in species as a whole in a very short time. We all have different sequences, but we have majority of it in common between all of us. So it gives you a lot of insight into evolution. And that ultimately tells you, okay, so how is the function changing? The genes are evolving. Is that having an effect on the function? Absolutely. Absolutely, it's having an effect on the function. Did you guys know that the enzymes in your body, all the enzymes that you have, they catalyze reactions, right? They make the reaction go faster, or they even make the reaction go. If we didn't have enzymes, if there was no evolution of enzymes, it would take millions of years for the reaction to occur in our body. Millions. For that one reaction that has to occur, it would take, say, 1 million years. But with the evolution of enzymes, that same reaction can be now done in picoseconds. So just have an appreciation for where we have come through evolution and what our bodies can do. There are enzymes in your body that have reached absolute perfection. I don't think they can evolve anymore to be better. They're absolute perfection. They have to be. There's an enzyme during your respiration that produces ATP that if there's something wrong with it, you will produce half the amount of ATP in your body. Half, literally, just cut in half. That's a drastic difference, right? So that enzyme has to be perfect. And how is that reflected? It's reflected in the genome. The sequence is different, which leads to a different structure of the protein, more advanced structure, you could say, because that's how proteins communicate. They change conformation, right? They bind to things. Their structure is critical. That's the bread and butter of their function. So that leads to change in structure, and so now they can perform certain things that they couldn't in the past. But all of that is evolving in the genome. It's reflected in the genome. The genome quantification also gives you an idea of how complex the organism is, quote unquote. Complexity, I put in quotations because complexity is debatable. In general, I think all scientists have accepted the fact that prokaryotes are less complex than eukaryotes, in general. Are there many metabolic pathways that bacteria can do that we can't do? Absolutely. But for the most part, single cellular organism doesn't have organelles, very simple genome. The smallest genomes that are found on Earth are that of prokaryotes, so they're considered relatively less complex. However, when you get to eukaryotic organisms, and especially multicellular organisms, which organism would you consider the most complex on Earth? You can raise your hand and answer. What organism would you consider to be the most complex and why? Yes? Platypus? Okay, that's the most complex organism. All right, good answer. I was not expecting that. I like that. Okay. Any other? Yes. Humans? Okay, why? Why are we the most complex? You guys are not going to become real scientists. You're going to debate this. There's no correct answer for this, but I want you to get into that mind of debating. Yes. Okay, the brain has more information, that's why. Yeah, Many scientists would agree that humans are the most complex because we have the most developed central nervous system. We're the most intelligent species. Therefore, we can quote unquote dominate Earth because we can use our mind to design things and to keep Earth the way we want to keep it, right? On the other hand, many scientists would disagree. You have to define the word complexity, right? In order for you to be able to say that we are the most complex organism. If complexity, by definition, is that you have a more developed and advanced CNS system, sure, that's great. But that's not what most scientists agree on, that that's complexity. What if the newt, or the axolotl, that can regenerate its entire limb, i.e. it can revert back to embryonic development in its adult body and develop cells that have nothing to do with each other from a lineage standpoint, right? Blood, muscle, bone, and it can regenerate an entire limb. Could the axolotl say, I'm more complex than humans? Yeah. If an axolotl could speak, it's probably laughing at us, going, you guys think you're complex. If I only had an advanced CNS. But what about a plant? What about species that can do aerobic respiration, anaerobic respiration, photosynthesis, all in one? Would they be considered more complex? What about organisms, right? Most organisms in the world that can split water. They can take water, the photosystem too, and can break it apart, i.e. it's very difficult, guys, to grab electrons from oxygen, which is highly electronegative. It will not give up its electron easily, especially just say, oh yeah, come split me open and take the electrons that you want. Well, photosystem too can do that. So would organisms that have that say that we're more advanced because we can split water and get sunlight and we're done? We can survive. We don't need to ingest food like you guys need to do, rely on all this, heterotrophic behavior. No, we're good. So, as autotrophs, yeah, we dominate. But anyway, just understand that the word complexity is in quotations. So if you ever have a debate with anyone, and you're going to sound intelligent when you really look at the science and understand what is complex, what is not, how we're evolving, what are the functions of these genes, what does the genome mean? I think a lot of these factors come into play just to understand the organism as a whole. And you sound really intelligent when you speak this way about an organism as opposed to, yeah, they're just like this because they are, right? You want to sound like, people are going to have a hard time debating you. So when we talk about the genome, what is the genome? The genome is all of the DNA in one copy of an organism's chromosomes. One copy is bold. So in your diploid somatic cells right now, your diploid cells, you have 2 copies of the genome. Your genome is only one copy, so the one from one parent. So you could say that your genome is the amount of DNA in your haploid cells. It's just the one copy. You have 2 copies of the genome. So where is this DNA? Well, it's in the nucleus. This over here is an example of a plant because I wanted to talk to you about chloroplast. The DNA that we'll be talking about and focusing on is a nuclear DNA. So when we talk about the genome of an organism, we're not taking into account the DNA in the mitochondria and the chloroplast. Did you guys know that you have DNA in the mitochondria as well, right? That is something you inherited from your mom, right? It's a maternal inheritance. So mitochondria and chloroplasts, they also have DNA. And they also have transcriptional and translational machinery. They have their own. And why is that? Why do they have DNA plus all the stuff they need to transcribe and translate? Why do they have that?
Because maybe like the mitochondria, like that came from prokaryotes.
Very good. They're descendants, right, of bacteria. There was a process called endosymbiosis that occurred many, many years ago, and this is where they came from. So they were once free living organisms. And now through the process of endosymbiosis, they're in the cell to make these organelles. So the mitochondria actually came from originally an anaerobic bacteria that could do aerobic respiration first. And so now that's what the mitochondria is, that's their main function, right? To produce ATP via aerobic respiration using oxygen as a final electron acceptor. Chloroplast also has DNA, and that first came from a bacteria known as cyanobacteria. Cyanobacteria was the first bacteria to be able to do oxygenic photosynthesis. What's that? It's doing photosynthesis by using water as the electron source, water. They said, why are you guys using things for electrons that you're having a hard time finding? Iron, sulfur, give me a break. You guys looking around, do you know how much water there's on Earth? Why don't we use water? So when they started splitting water, they started releasing oxygen as a byproduct. And that's how you got the evolution of oxygen in the environment, right? The atmospheric oxygen came from that. So during that time, you had cyanobacteria doing oxygen photosynthesis, and boom, endosymbiosis occurred, and now here you go, we're here in 2025, right? So understand that there are DNA in other organelles as well, but when we talk about characterizing the genome, and we're talking about all these copy numbers and whatnot, we're focusing on this. There are many mitochondrial diseases that I may talk about next term, and they focus on the mitochondrial DNA. So we're talking about karyotypes, right? This is happening during metaphase. This is the chromosome. This is the centromere, et cetera. This is the blob that you guys see often, right? It's this big gray, black blob. That blob is a chromosome. And this is the actual picture. It's not a cartoon picture. This is why it looks like that. I want you to understand what that blob is. To understand this, you have to understand this. This is what the DNA structure looks like. It's a double helix. It has polarity and antiparallelism. So if this end is 3', you follow this, the other end is 5', and vice versa. So a 3 and 5 have to go together. Don't put a 5 and 5 and a 3 and 3 together. 5 and 3, 3 and 5. Okay. When S phase occurs, this is during G1, when S phase occurs, then you get duplication. We'll learn about DNA duplication next class, but through semi-conservative replication, you get the production of 2 double helices. Both of these come together, right, and they form the sister chromatids. When your chromosome duplicates, it looks like this. This is the chromosome, that's the chromatid. That's the chromatid. So if I ask you in this entire image, how many 5' ends are there, the answer is going to be 4. 2 from this strand, 2 from this strand. The answer is not 2, the answer is not 8, it's 4. So understand what that blob means. What are we looking at in terms of DNA structure? Just a quick recap on the definition of a chromosome before we hit into ploidy and numbers like that. You have one chromosome, it gets duplicated, and it looks like this with sister chromatids. That's still one chromosome because it's one centromere. When it splits up, right, during anaphase and telophase, when it splits up after cytokinesis, you get still one chromosome in each because you still have one centromere. So one chromosome, one chromosome, one chromosome. Now let's get into the numbers. N value and coefficient of N. N value is the number of unique nuclear chromosomes present in an organism. And just before I start this slide, I know I mentioned this is one of the more difficult lectures, but just understand that if you get these definitions and if you do some practice, you should be perfectly fine. The confusion happens when you don't understand this lecture. So just if you're going to pay that extra little attention, this is the time. All right, so N value is a number of unique nuclear chromosomes present in an organism. So how many unique ones do we have? We have 23 unique ones. All right? Don't look at the X and Y and say, but isn't that 24 unique ones? That's a technicality we're not going to touch. Just XX or XY, we just consider them homologous, even though they're not. The XY are not homologous, we know, but for purposes of characterization in terms of numbers, we consider them homologous. Coefficient of N, that is your ploidy, the number that comes beside the N, that's the ploidy. That tells us the number of unique sets that we have. So we have 2 sets, our ploidy is 2. Those that have 3 sets, the ploidy is 3, 4, 5, 6. There are plants with 6, 7, 8 ploidy, right? This is one homologous pair. The sizes are the same, right? One homologous pair. This means there are 2 sets of one unique chromosome. The unique chromosome is right there. This is not unique. So You have one copy from mom and one copy from dad of chromosome 1, for example. Those are not considered unique. Even though you're like, well, wait a second, the sequence could be unique, it probably is, right? Those are just different alleles. The genes are in the same loci. The sequence is roughly the same. But yeah, there are differences, right? Because you get different alleles. They could be the same allele or different alleles. The bottom line is uniqueness only comes when the chromosome number changes, right? 1 and 1 are homologous. That's not unique. So this is the unique one. So in this case, 2n equals 2, where n equals 1. Where n equals 1 because you have one unique one. So this is a diploid. Now let's talk about C value and coefficient of C. C value represents the amount of DNA in one set of an organism's nuclear chromosomes. That's known as the genome size. So it's not how many chromosomes do you have in one set. My question here is, what is the weight or the length of that set? That's the genome size, or C value. So do you guys know what the C value for humans is? The C value is measured in either base pairs. It's measured in either base pairs or in picograms of DNA. So how many base pairs do we have? Yes. 5 base pairs? Yes. ACTG, yes, we have it. But how many of those ACTGs do we have? Yes. 10? 10 in each? Okay. Like 10 base pairs? Yeah. Okay. Do you know how many we have? 3.2 billion. 3.2 giga base pairs. 3.2. Giga is billion. K, kilo, M, mega, million. And that's roughly 3.3 picograms, okay? 3.3 picograms. So if you took one set and you weighed it, it would weigh 3.3, yes. So that's your genome size, guys. When I say genome size, people go, what's genome size? I didn't know what the genome size was on the exam. I didn't know what you were asking. I was asking for the C value. Oh, I know the C value. I just didn't know that C value meant genome size. That's what it means. The coefficient of C tells us how many times the entire genome is present in the cell. That's the copies of your genome. How many copies of the genome do you have? Do you have 1? Do you have 2? Do you have 3? Copies of the genome will be different as the cell cycle progresses. This is also known as copies of the genome. So coefficient of C, copies of the genome. So write that down, copies of the genome. When I ask you on the exam, you need to understand what they mean. I won't just say, what is the coefficient of C? I may say, how many copies of the genome are present in this cell currently? So you would have to know what you're calculating. Here you have another homologous pair. Each chromosome is duplicated. Therefore, the entire genome is present 4 times, guys, because this is the unique sequence. It's just the one copy. This is not the genome, and that's not the genome. Only this strand here, that chromatid, represents the genome. So there are 4 copies, not 2 copies. You take this, and that's 4 copies, because this is the unique one. This is just duplicated, right, guys? This is a sister chromatid. But these are homologous, so they're still the same one. So they're not unique. So just count that one. That's 1. So let's do some examples. Identify the ploidy and N value of this organism. Let's say there are 2 sets of homologous triplets that make up an organism's genetic material. Here you have 1 unique chromosome copied 3 times. This is a triploid organism, for example, the seedless watermelon. This here is a 2nd unique chromosome copied 3 times. On the exam, the way I'm going to tell you if chromosomes are unique is by differences in length. If I draw them different lengths, you know on the exam that these are different chromosomes, and that will help you determine the N value and the ploidy. So here you have, how many unique chromosomes are there? Just two, that size and that size. Put that in one set. So this is one set, another set, another set, right? This is not a set, and that's not a set. Sets have to have unique ones in them. So therefore, the number of sets of unique ones are 3, not 2. Another common mistake. So just pay attention to what a set means. So this is a triploid, 3N organism. There are 2 unique chromosomes, here and here, 3 times. So N equals 3. So 3N equals 6. So a common mistake would be the following. 3N equals 6. Yeah, you could say, yeah, I can count the number of chromosomes. The total number is 6, sure. And you put 3N, right, where you would say, you know, N equals 2, that's correct. But some people would put 2N equals 6, where N equals 3. That is incorrect. And you all know that's going to be one of the answers, right, on the multiple choice. So just make sure that you understand those nuances. Identify C value and coefficient of C. Now we're at coefficient of C and C value. What is C value? The genome size and then copies of the genome. When we talk about copies of the genome, pretend like you're photocopying a piece of paper, an 8 by 10 piece of paper. That's your genome. That's your actual C value. That's not going to change in length, right? The original copy. You stick it on the photocopier, you close the photocopier, you press a button, and it makes copies. The copies coming out are the coefficient of C. The paper, the original paper in the photocopier, is the C value. So don't change the C value. Important thing to note, C value of a given species remains constant. It's always constant. It's not going to change. So when I ask you, how is the C value changing throughout the cell cycle, you're going to say it's not changing. It's the same. The copies of the genome are changing, but the C value is not changing. When that paper's in the photocopier, no one's picking it up, cutting paper out of it, putting it back, or sticking paper, putting tape on it. No one's doing that. That's the copy. It's done. What's changing is how many copies you're making, right? So keep that in mind. Let's say that these are homologous triplets again that make up an organism's genome. So here you have triplet, triplet. This right here on top, that's part 1 of the whole genome, duplicated on each chromosome, copied 3 times into homologous chromosomes. This is part 2 right there. That right there is part 2 of the genome, duplicated on each chromosome, copied into 3 homologous chromosomes. So this, you guys, right there, that and that put together is your whole genome, part 1 plus part 2. Each part is present in the nucleus 6 times. So right now it's sitting as a triploid organism, 3N, but the copy number is 6C. Okay, so the copy number is 6C, so it's 3N, 6C. Therefore, you have 1, 2, 3, 4, 5, 6, where C is the total mass in picograms. I could give you the total mass of this whole thing and ask you to calculate the C value. So I would say, what is the C value? The way to calculate that, I've given you the value of C, right? This is math, guys, math. I've given you the value of C. You have to tell me what the total is, or I could give you the total and you tell me what the value of C is. Do you know what the secret to that whole calculation is, what I'm looking for? I'm looking to see if you know the coefficient of C. If you know the coefficient of C, you will know what to multiply it with, or you will know what to divide it with. So what I'm actually looking to see is if you understand what the genome copy number is. And, you know, on the multiple choice, the options will be, did they divide it by 2? Did they divide it by 6? Did they divide it by 3? Or did they divide it by 12? All those options will be there. So if you see your correct answer, go, there you go, there's my correct answer. Just double check your work, because the answers are there based on what the possibilities of division could be. I'm not going to say, well, what if they divide it by 7.6? No one's going to divide it by 7.6. People are going to divide it by either 2, 3, 6, or 12, right? A trick, you guys, a trick is to count the number of sister chromatids in each homologous grouping, and that's your coefficient of C. So just take one homologous grouping in the entire karyotype and just count all the sister chromatids. 1, 2, 3, 4, 5, 6, done. That's a trick. But if I teach it to you like this, you won't even understand what a genome is, right? Or what a copy number of a genome is. But that's a trick. Once you understand the basis, then understand how you can use little tricks to come up with the answer. In the past, I used to use the box of chocolate as an example in office hours, so students came to me and said, you should use this example in class. So I did this example for the last 2 years, and in general, students came up to me and said, it wasn't super helpful, but it was okay. So let's just run through it really quickly. I'm not gonna spend too much time on this. Here's a box of chocolates, 6 different flavors. They're different, right? So n equals 6. But this box is still one box, right? So it's only one set of chocolates, one set of that 6 different flavors. It's one set of box, so that means that the ploidy is 1, n equals 1. If you took all the chocolate and weighed it, and say it weighed 200 grams, that would be the C value of this box of chocolates. This one box weighs 200 grams. The weight of the one box will never change. It'll always be the same. If you have 2 boxes, that's not going to say, oh, since you have 2 boxes, that means on the weight of the first box is going to change, the weight of the second box is different. No, it's still 200 grams. Each box is still 200 grams, and that's the C value. Our C value is always 3.2 billion bases, right? Or 3.2 or 3.3 picograms. If you have 2 boxes, now you have a diploid. So you have 2n, 2 sets, 2 boxes, 2 sets of chocolate. So you have 2n. It's a diploid. But the total number of chromosomes, or the total number of chocolate you have, are actually 12. But you only have 6 unique ones, so n still equals 6. You only have 6 unique flavors. You don't have 12 unique flavors here, right? The C value is still going to be 200 grams. The one box of chocolates. No one wants to know how many grams you're carrying in your hand. They just want to know what's the weight of one box. I will calculate the total grams based on how many boxes you're holding. Got it? Third box, 3n, 3n. N still equals 6. Unique number of flavors are still 6. The total, though, is 18. 3n equals 18. This is a 3n equals 18 organism. C value, 200 grams still, right? And the copies of the genome are going to coincide with n. So in this case, it's C, 2C, 3C. The copies of the genome are the same as a ploidy during G1. This one is hopefully obvious again. 4n equals 24, C value 200 grams, and coefficient of C is 4. Was that helpful, or was this like not so helpful? I don't know, I can't tell. It's a 50-50 split. Some people think I wasted their time. Some people, if you think this is a waste of time, please ignore this slide and just move on with whatever else. All right, so human karyotype. An important thing to note here, very, very important. Mistakes are made when this concept is not understood. If this is in metaphase, and I ask you, how many copies of the genome do you see in this figure right now, in this karyotype? How many copies are there? My question is simple. How many copies of the genome are present in this karyotype? Yes, 4, okay. You're saying 4 too? How many agree with 4? How many say 2? Okay, okay, so there are actually 4 copies of the genome. Count the number of sister chromatids. You know how you come up with that? What is this blob right there? What is that? They're sister chromatids. Pay attention. The resolution when taking the photography is not really high. There are pictures that have higher resolution where you can see the sister chromatids, but I'm not going to present that to you because that's not the norm. Normally it's presented like this because that's how it's seen under the microscope. These guys have to be sister chromatids because it's taken during metaphase. These are sister chromatids. So take that right there, this one strand, and draw sister chromatids, and take this and draw sister chromatids right now in your notes so you understand that there are 4 sister chromatids in this homologous grouping. So remember, the trick was to count all the sister chromatids in one grouping. That's your coefficient of C. All right? And honestly, guys, another trick, just count the number of centromeres in one homologous grouping, and that is your ploidy. And to get your n value, just look at all the numbers, count them. Right now they're 23, right? But if I don't label the numbers, you physically just count them, right? Count them, 1, 2, 3, 4, 5, 6, you physically count them yourself, and then that's your n value. Your C value, I always have to give you. You can't come up with the C value. How do you know how much the DNA weighs or what the length of the base pairs are? You don't know. So I will give you that value. So I'll either give you the C value and tell you to calculate the total mass of this karyotype, or I will give you the total mass and tell you to calculate the C value. So let's go through the amount of DNA or the genome copy number in a diploid cell. It's a diploid cell. So in a diploid cell, what are you going to start off with in G1? You're going to start off with 2C. What's going to happen during S phase? Is the genome copy number going to go up, down, or stay the same? Up to 4C, very good. I'm hearing a lot of 4C, good. And what about in G2? Up, down, same, it stays the same, very good. And what happens at the end of M phase? It goes down. It goes back to 2C when the cell divides. So after cytokinesis, it goes back to 2C. Don't worry about any technicality during anaphase. There's a technicality that happens during anaphase where you get more chromosomes, more centromeres. We are not worried about that. No one cares about the ploidy and the genome copy number during anaphase. Okay, that's not important. We want the end result. The end result is that you go back to a 2C. You have to go back to a 2C. Now let's model the ploidy in a diploid cell over the cell cycle. The ploidy, this is no longer the genome copy number. This is the ploidy. Keep that in mind. Coefficient of C and coefficient of N are two completely different things. So in a diploid organism, during G1, it's 2N. During S, what is it? The DNA replicates. It's still the same. Tell me why it's still the same. You are correct. The ploidy is the same. It does not go up. But you're like, but the DNA replicated. So how come the ploidy is not 4N? Why is it still 2N? It has to do with chromosomes, yes. Chromosome number, yes. Good. It's the centromere definition, right? The same number of centromeres. We're not counting the sister chromatids as different chromosomes, right? When they're stuck together, the centromere number is still the same. So absolutely, yes, remaining the same. Ploidy is constant throughout the cell cycle. Here's a cell. It's a diploid, right? So you have a chromosome here. These are all homologous, right? And they're supposed to be different sizes, so just bear with me if they're not different sizes, but these are all different sizes. So these are two sets of chromosomes. They're diploid, and you can see 8 of them in total. Each one of these will have a centromere, correct? Each one of these will have a centromere. Now, when the DNA replicates, take each one of these and just make them sister chromatids. Each one of that and just make sister chromatids. Do you notice that the number of centromeres are the same? So do you still have 2 sets? You sure do. You still have 2 sets of chromosomes. That's why the ploidy doesn't change. N implies nothing about C. What this means is you can't look at the N value and go, oh, I can predict the C value. I have a pretty good idea, rough idea. No, there's like literally no correlation. Look at this. The cucumber versus the Tasmanian devil. The cucumber has 7 unique chromosomes. It's a diploid, so the total number of chromosomes is 14. So does a Tasmanian devil. It also has 2n equals 14. It is a 2n equals 14 organism. So we mathematically equate these organisms, right? We are 2n equals 46 organisms. So 2n equals 14. But look at the C value. The C value of the Tasmanian devil is so much larger, pretty much the size of ours. 3.3 billion base pairs as opposed to 243 million base pairs. So what can you deduce from this? Just by looking at these numbers alone at the bottom, what's one conclusion you can make about the chromosomes of the Tasmanian devil? Yes. They're longer, absolutely, yes. They're significantly longer, right? Maybe not each and every one of them is significantly longer, but a good proportion of them has to be significantly longer in order to account for that 3.3 billion bases, right? So the Tasmanian devil packs in so much more DNA in the same number of chromosomes than a cucumber. N implies nothing about C. In this case now, the C values are very similar, but look at the difference in the chromosome number. The Tasmanian devil versus the human. 2n equals 14, 2n equals 46, very similar number of bases. We have the same amount, roughly, as a Tasmanian devil, but it can do it and pack it in 14 of them. How much DNA do you have in your nucleus of your liver right now? If you take one cell of your liver, how much DNA, if I did it in base pairs, how much DNA would I be able to find in there? You guys have to be able to do this on an exam. Come on, you can do it, guys. It's 10:13, we can do this. Guys, I told you the genome size. We're diploid, and you guys should know what the liver is. Liver is not your egg and sperm, right? Liver is your diploid cell. So how much DNA should I see in the nucleus? If I were to weigh it, or if I were to measure it in base pairs, yes? Very good, 2C. So it would be 6.4 giga base pairs, or roughly 6.4 or so picograms. Very good. You gotta know the coefficient of C to calculate that stuff. N implies nothing about C. Here you can see a vast difference in the C value. Look at the axolotl, guys. The axolotl has 32 gigabases in its genome. In 14 chromosomes, it packs in 10 times the amount of bases that we have. Isn't that incredible? Why? Why do you think the axolotl has 10 times more DNA than us? What would be like a hypothesis that you would say, okay, you know what? I would hypothesize this as a scientist, yes. Okay, because it can regenerate. Do you think it needs 10 times more to regenerate? Do you think all that DNA is sequencing for RNA and protein? Yes. Oh, that's a really good one. I like that one, that there's a lot of gene duplication. That would be an awesome theory that you would have to test out, right? Very good, I like that. There are gene duplications in various organisms. The answer is, we don't know. The answer is, we don't know why the axolotl has so much DNA, okay? Because most of the genes that it uses to regenerate, it already has used in the past through embryogenesis. It's not using a whole array of different gene expression patterns. It's just using very similar ones. There might be a few extra here and there, but certainly not to account for 10 times the amount. So why is it packing on so much DNA through evolutionary time? Plants do the same thing, guys. So this leads us to the C-value paradox. C-value paradox says that the C-value does not correlate to the complexity of the organism. So you can't say the higher the C-value, the more complex the organism, unless you're comparing prokaryotes to eukaryotes. There I can say, you can say that. Look at these bacteria, such small C-value, right? And they are far less complex too, they're considered less complex. But they're much easier to work with in the lab. That's why we all work with bacteria in the lab, right? They're much easier to manipulate that genome, small genome size. Mammals, we're right here. We're somewhere here. We're obviously not the only mammals in the world. There are many mammals, and so mammal range is here. The range simply means that this is the range of C-value that you will find in different species in the world. Look at plants. Look at plants. Highest C-values seen on Earth are those in plants. Here you have the Paris japonica. Its C-value, guys, is 150 giga base pairs. Ours is 3.2. Put that in perspective. Ours is 3.2. This plant has 150 in its genome, And these guys are like, tetraploid, hexaploid. Can you imagine the amount of DNA and the replication process? Isn't that insane? So the question is, why are they packing it on through evolutionary time? 85% of sequence in the DNA of plants is transposable elements. We'll talk about that in subsequent classes. They're just there for the sake of being there, right? They jump around. The point is that plants pack this on through evolutionary time, and we really don't understand the meaning behind it fully. Look at this marbled lungfish, C-value 130. Do you think the marble fish is more complex than us, or is this Japonica plant more complex than this? Is there any correlation you see here? No, right? There's no correlation. And the axolotl 32, as you know. This slide here is a beautiful summary of how I will test you. I'm not even supposed to put this summary up. I'm supposed to tell you the C values and all this N values, ploidy, etc. Couple that with mitosis and meiosis, and then ask you on the exam, tell me the ploidy, tell me all these numbers of what's happening during anaphase 1 or anaphase 2. That's the expectation at the university level, but what I'm doing for you is I'm basically telling you, know this. I'm giving you the biggest hint of the world, is to say, no, this is how I'm gonna test you, understand it. So it's just very, very straightforward. Don't overcomplicate this. If you have questions, do come see me, but I'm giving you a hint as to how this will be asked. Watch my videos on Brightspace. I've talked about this. Let's do this practice. If you have this 2 gametes, and if you then produce a zygote, let's go through this. You guys know what the zygote will be, right? Okay. The resulting zygote will have a ploidy of 4n. Is it true or false? False. What is the ploidy of the zygote? 2n. The ploidy of the gamete is 2n. The gamete that you see, is that 2n, or is that n, or is that 4n? What is a ploidy of the gamete? N. 1 unique set. Count all the different sizes. That's 1 box of chocolates. N. So these are n. The 2n come together to make 2n. The gamete has 4 copies of the genome. No, it has 1 copy of the genome. Count all the unique ones, that's 1 copy, okay? It has 1 copy of the genome. This is a 2-step, number 4. The resulting zygote will have 4 copies of the genome during the G2 phase of the cell cycle. To calculate this, first ask yourself, how many copies during G1? Multiply it by 2. So how many copies during G1? The zygote, 2. This is a 2N, 2C zygote. Just multiply it by 2. Yes, it will have 4 copies. So this, in fact, is true, and the rest are all false. So in a multiple, this is literally out of an exam in the past, guys, literally. The answer is 4 only. The answer is 4 only. If you go to my test your knowledge and sample question video, I take this question and I rehash it like crazy. And you will learn everything you need to learn about this topic with this question. The way to do it is to take sister chromatids and change it to chromosome. Take G2, change it to G1 or S or M, and take the drone, that's a haploid, and change it to the queen bee, that's a diploid. You will have beautiful combinations to work with, and the answer keeps changing each time. And this was a great exercise.